We are about to go on a rather unusual journey, a stroll around the unknown universe of invisible life. To do this, we'll need a microscope, in order to enlarge images hundreds of times over. The inhabitants of this universe can be measured in terms of a few tenths or even hundredths of a millimeter, and are made up of a few hundred cells or even a single cell. Compare this to the billions of cells that make up a newborn chick. We will be surprised by the wide variety of shapes and the strange habits of these creatures, which predate our own arrival on Earth by hundreds of millions of years. They managed to survive a series of catastrophic events and witnessed the upheaval of continents and the birth and death of gigantic species such as dinosaurs. Over the years, many of their orders disappeared without trace. But we can still find traces of many of them in the darkest depths of the sea and in the remotest regions of the world. Even if differing in proportions, their DNA is similar to ours, or rather, it is us humans that inherited from them a basic common molecular structure. For this reason, these creatures today represent a priceless source for experimental research in genetic engineering. We will obviously not be dealing with this subject. We are only interested in the random observation of the behavior of the inhabitants of this fascinating universe, the universe of the infinitely tiny. There are many available sources of observation: puddles, ponds, patches of moss, and even the water where our cats and chickens drink. Let's start our journey. Every drop of water which is placed in the indentations of the slides will be like a sea, measuring only 8 millimeters. Our voyage starts with bacteria. We know some immobile types which have a number of different shapes. Contrary to what we might think, bacteria already possess a complex structure. For instance, in order to protect themselves from viral DNA, their worst enemy, they are endowed with particular enzymes which recognize and fragment every foreign DNA, whilst at the same time defending their own. Many bacteria, on the other hand, are very mobile and reproduce every 20 minutes. If they were not to encounter enemies, they would cover all dry land in a very short space of time. Although they have been on the Earth for more than 1 billion years, it is believed that they were preceded by even simpler microorganisms now extinct. They are to be found everywhere, and live with us and inside us for all our lives, and devour us when we die. But bacteria have so many natural enemies on this planet that there is no risk of this happening. Among these natural enemies, we find the infinitely large army of ciliates, about which we shall speak later. Let's follow them for a moment in order to see just how much effort they put into their relentless hunt for bacteria, which they draw into their mouths with the tireless motion of their cilia apparatus. Now let's watch some sequences which represent a sort of tiny ecosystem wherein all parties, the hunted and the hunters, are guaranteed a stable existence. Inside this ball, which measures a few tenths of a millimeter, we can find thousands of tiny microscopic creatures. They are bacteria. All of a sudden, the ball explodes, and this occupying army is scattered into the immediate environment. They are going to set off to hunt a host, maybe large-scale prey such as mammals and humans. But hardly have 30 seconds gone by when a ciliate pounces on this teeming multitude like a thunderbolt. Let's watch the scene again. Let's follow it for a while as it goes backwards and forwards like a soul in torment. In the space of a few minutes, it devours an astonishingly large number of bacteria. We can see it right under our eyes as its size increases, and while it gets darker and acquires a jagged shape that it previously did not have. It will only call a halt to the slaughter when it has literally died of indigestion. So here we see it motionless, lifeless. Another minute goes by, and, lo and behold, yet another diner passes by. This is a small amoeba which initially is happy just to capture single bacteria, before attempting a bigger project. It goes up threateningly to the ciliate which has unwittingly become an enormous storehouse of fresh food. It attempts to swallow it up, but the prey is too large, and so in the end it gives up. At this point we must ask ourselves what type of message was sent from that multitude of bacteria to their predators. What signal was sent at the time when the ball exploded and that brought the event to the attention of these hunters who previously had not been present? And here is the resplendent heliozoa. It consists of a single cell, and its rays remind us of those of the sun, falling down from the sky into a drop of water. But these rays, known as axopodia, are lethal instruments. By means of these rays, heliozoa introduce into their prey a substance which paralyzes them before absorbing them. The victim can be as large as 0.5 millimeter in size. Heliozoa can sometimes be observed in groups, albeit instantaneous. It's possible that about 500 million years ago, these pluricellular beings originated from unicellular groupings, which did not then pass on to the separation phase. Diatoms are a few hundredths of a millimeter in size, and have been living on our planet for over 100 million years, both in fresh and in seawater. They have silicate structure with very differing shapes. Over the millennia, their remains have formed sedimentary rocks known as diatomaceous earth. The ones we can see here look like tiny submarines. Some marine diatoms are of fine circular shape. This unusual marine diatom, a few hundredths of a millimeter in size, is known as meridian circular and looks like a fine fan. Ciliates are unicellular protozoa which have achieved a remarkable degree of evolutionary development. The cilia, from which the name of the class is derived, enable them to move around rapidly and in all directions. Ciliates measure a few hundredths of a millimeter and multiply both by means of direct division and, as we shall see, via coupling and exchange of their DNA. These fine bell-shaped vorticella are ciliates from the peritrichal order, and they contain a perioral corona of cilia. They often live in colonies and feature a long retractable peduncle. Flagellates are so called because they are endowed with one or more very mobile flagella, which are used to capture and absorb food, which is mainly composed of bacteria. Like ciliates, flagellates consist of a single cell, and are of a few hundredths of a millimeter in size. Here we can see a specimen with a single flagellum, which could easily be confused with some unicellular algae which also have a single flagellum. Flagella also feature in some algae, such as these dinoflagellates, called horned algae because of their shape. An interesting fact for biologists. The chromosomes of some dinoflagellates are also recognizable in nuclei in interphase, in other words in cells which are not undergoing division. A common feature of all algae is autotrophism. In other words, they are cells which can extract from sunlight energy necessary to transform simple inorganic molecules into complex organic structures. Unicellular algae are a few hundredths of a millimeter in size and have a number of very different shapes. They can live singly or in colonies. Some algae have beautifully symmetrical shapes, such as this one from the order of Chlorococcales, which resembles an 8-point star. The order of amoeba is represented by unusual unicellular beings with no definite configuration, and which continuously change shape as they move. They consist of a thin, transparent external layer and of an internal granulated one. Their movement is made possible by the flow of their contents, which are propelled in a forward direction, thus enabling them to move, as we can see in the case of this half-millimeter amoeba. This is called amoeba proteus, on account of its protiform appearance, as we can see in the following series of accelerated images. Biologists will be interested to know that the nucleus of this cell is that small, dark, round mass which is visible near the center of this tiny animal. It has more than 500 chromosomes. Thick amoebians are a few tenths of a millimeter in size. They possess an extremely tough shell, which also contains extraneous siliceous particles. Their crawling movement is due to pseudopods, which vary in shape from species to species. These metazoans introduce us to the world of microscopic pluricellular beings. Rotifers are so called on account of the coordinated movement of the corona of perioral cilia, similar in appearance to two rotating wheels. Around 2,000 species of these tiny animals are known, the largest of which can be up to 1 millimeter in size. The movement of the two wheels is a fascinating sight. They possess an apparatus for sucking or chewing, a dilating and contracting stomach known as mastax, originally thought to be the heart, and a digestive system and kidneys. Rotifers consist of a few thousand cells and do not live for more than a week. While the previous specimen was from the monogonant order, we are now looking at one from the Brachiones family. To the sides of the contracting stomach we can also see the kidneys, which appear as two small clear masses. This is a fine round testudinella of vitreous appearance. The tiny animal can measure from 120 to 200 thousandths of a millimeter. Here the pulsating mastax is clearly visible. Here we can see a rotifer from the Phylinia genus, with two very long aculeate brushes which measure about half a millimeter. The profile of the cilia corona of rotifers from the Floscularia family is undoubtedly very striking and reminds us of splendid tropical butterflies. Tardigrades are tiny animals of unmistakable shape and behavior. They have been living for hundreds of millions of years in all regions of our planet, and have been found as far away as the Antarctic. Their place among the metazoan order is still uncertain. They move slowly, hence their name. They measure a few hundredths of a millimeter, although the largest can be up to half a millimeter in size. They possess 4 pairs of legs, each endowed with 2 nails used as claws for the purpose of climbing. This is a tardigrade found in moss at the foot of a tree on Monte Terminio near Avellino. It's from the Escinius family and has 4 pairs of long cirrus. Tardigrades have a mouth provided with sharp dagger-like features used to perforate the walls of the vegetable cells on which they feed. Nematodes make up the class which probably contains the most species. They are thought to be around 100,000 different species of nematodes, which differ only in a few tiny details. They can measure up to as much as 1 millimeter, and they move in a rapid snake-like way which makes it difficult to observe them. Annelids are tiny terrestrial or aquatic animals whose bodies are divided into segments. What we see here is Scolopendra marina. It's a few millimeters long and has 21 pairs of appendages, just like the terrestrial ones, which can measure up to 10 centimeters. We do not know their evolutionary history, But we might suppose that a few million years ago some of these microscopic scolopendra decided to abandon the sea so as to adapt themselves to a terrestrial life. These microscopic annelids belong to a single genus. They are of varying lengths and are made up of segments which are not clearly delineated. A long annelid which belongs to the same family as the previous one. This annelid has a disc-shaped reddish egg in its abdomen, and indentation whose importance we shall shortly explain. The category of platyhelminths or flatworms includes many classes of metazoans, including Turbellaria, trematodes and cestodes. They are of greatly differing sizes. The one we see here is a Turbellaria measuring a few millimeters, with cilia and two black eyes set far apart. This flatworm does not have cilia, but has two sharp spines. This specimen has cilia and is of a fine light blue colour. It could be confused with a large ciliate. This flatworm is a few millimeters long, with small wide-set eyes and a cilia apparatus which it uses to draw food into its mouth. This is another specimen from the same family. In its abdomen we can see a large egg ready to be deposited. This flatworm, with a peculiar triangular head, has two close-set eyes, which give it the appearance of being cross-eyed. For millions of years the sea has been home to an infinite number of microscopic mollusks, many species of which still have to be discovered. Some are lacking in a shell, such as terrestrial slugs, while others have a protective shell, such as snails. The ones we can see here are microscopic marine mollusks. They are extremely mobile and have a large number of cilia. This is an unusual marine mollusk without cilia, and with a peculiar way of feeding. This highly unusual species of octopus lives in the sea and is not larger than 2 millimeters. From its apparently amorphous body there emerge 5 mobile tentacles, each of which terminates in a pair of suckers which the animal uses to attach itself to the ground. This marine mollusk is a couple of millimeters in size. It is similar to the clams which we farm and eat. Its evolutionary history appears to have preserved structures and biological models not unlike those of their ancient forefathers. There is a really surprising similarity between this microscopic sea snail and its terrestrial sibling, which we find on our lettuces. It is not always an easy task to make a detailed reconstruction of the route undertaken by these Lilliputian beings from life in water to that on land. There must have been an intermediate stage of life, as we can see from amphibious gastropods, which live both in water and on land, provided there are high levels of humidity. Brachionous rotifers are drawing food into their mouths with extraordinarily coordinated movement of their perioral cilia. Rotifers belonging to the Trichocerca genus, such as the specimen we can see here, are endowed with an oral apparatus which lends itself to grabbing movements. The Coloteca rotifer has long brushes which draw tiny algae into its mouth. The algae are then swallowed and transferred to the throat. This unicellular creature is moving up and down inside a dead rotifer, as if it were trying to find a way out, like an embryo which has become trapped inside its dead mother. Although in this dead tardigrade the occupant looks immobile, it is not unlike the previous example. Here we can see a number of multiplications, which in the end completely fill the dead body. These particular formations are not embryos or parasites, as we might have thought, but a kind of scavenger, insofar as they are feeding on corpses. Here we can see a crowd of them busying themselves on some dead rotifers. Here a scavenger is trying to gain access to a dead rotifer, which has in part already been occupied by other participants. In the end, it succeeds. Here we can see another scavenger easily gaining entry to another dead rotifer. When the banquet is over, the participants get ready to depart one by one, after they have devoured all the tissues, leaving only the outer shell of the corpse. Here we come across scavengers from the same family as before, while they feast on the remains of a long annelid. We can also find instances of real cannibalism among rotifers, as in this case. This rotifer has an enormous egg in its abdomen, almost ready to be deposited. Let's follow it for a while. After about 3 hours, the egg is laid, but the mother does not leave the embryo until it hatches. The first signs of movement appear in the embryo. In a few hours' time it will hatch. Now a rotifer from another family stops near the embryo and then goes away. A few seconds later, the predator rotifer returns and attacks the embryo with biting and sucking movements. The embryo is still alive, as can be seen from its interior movements. The predator then goes away, and the corpse of the embryo is attacked by a host of bacteria, while the mother remains completely ignorant of what has happened. But the story is not yet finished, because, after a while, the predator comes back and starts wandering around nearby. It is still not full, and gives a few more bites to the remains of the embryo. And then, all of a sudden, it attacks the mother. Then finally it goes off for good. We have seen one of the many struggles for existence carried out instinctively in this case by a tiny animal a few hundredths of a millimeter long. But cannibalistic phenomena also take place in the universe. These are two galaxies which are about 300 million light years away and made up of billions of stars. In the space of 1 billion years, one of these galaxies will take over the other, thereby producing unimaginably catastrophic events. Tiny microscopic animals also need to rid themselves of food they have digested, and they do this by bringing it out by a means of a series of coordinated movements. A brachionoid rotifer is here discharging its feces. Here is a floscularia which is expelling its excrement. What is unusual is that the excrement is always expelled in the same direction. We can see that it is all gathered in one place. Here is an annelid with excrement which has just been expelled from its intestine. Microscopic animals' movements and locomotive systems are many and varied, and some are highly unusual. In monogonont rotifers, motility is said to be looping, and resembles that of the caterpillar of the pine processionary moth, rightly called a looper caterpillar. The Tecamabina has a crawling movement on account of its lobose, branch-like pseudopods. We see a flatworm or large ciliate. All of a sudden, it starts making rapid rotatory movements in a rightwards direction. It's like a real spinning top. Here rotation is in a leftwards direction. In the few moments in which it pauses, we can see that this skillful skater is a ciliate, a few hundredths of a millimeter in size. The smooth, coordinated movements of these diatoms are unusually mysterious. They have been filmed here at varying magnification. The movement of these splendid microscopic vorticella is fascinating. They seem to be twirling in some unknown sky. We know that sloughing is the process whereby animals such as snakes change their skins. But also tiny microscopic animals shed their skins. What we see here is the shedding of a tardigrade skin. The process will take a couple of hours and will leave an empty cuticle known as exuvia. Here we see a tiny nematode with its exuvia. Here is another nematode next to its exuvia, which has just been cast off. Here is an exuvia which has just been cast off by a tardigrade, a few millimeters away. Rotifers do not cast off their skins during their week-long lives. What we can see here is not the exuvia of a brachionos rotifer, as we might think, but its exoskeleton. The chances of survival for waterborne microscopic animals are very low. Survival is not only ensured by their being highly prolific, but also by some special features such as hibernation when water is scarce or absent. Here we have gradually dried out a drop of water in which this rotifer lived. The animal starts to drink water so as to guarantee itself plentiful reserves of water during the forthcoming emergency. Here it takes on the appearance of a static sculpture. After a couple of days, we pour a drop of water onto the strange form that this animal has become. Let's see what happens. After about an hour, it will go back to being as before, as these non-real-time sequences show. This tardigrade has been taken from a piece of damp moss and placed in a drop of water. We dry the slide out slowly. First of all, the tiny animal provides itself with a supply of water, and then remains completely static in the dried out area, as if lifeless, similar to a mummy. The following day, we pour a drop of water onto this arid region, and the mummy starts moving. After about an hour, the tardigrade returns to normality, albeit slowly. We have witnessed these unusual capacities both in rotifers and tardigrades. Other species, such as nematodes and flatworms, are not able to do this. The hibernation technique adopted by tardigrades in nature is quite different from that which they have to carry out rapidly in an artificial context. When water disappears from their natural habitat, tardigrades retract their head and claws into a sort of barrel where they can stay alive for very long periods of time, months and maybe years. When they come into contact with water, these little barrels, which belong to these ancient inhabitants of our planet, swell with water, and in a few minutes the animal returns to its original state. These sequences have been filmed in non-real time. Recently, an interesting discovery has been made. These little barrels can survive cosmic rays. They have been placed inside spaceships and exposed to cosmic rays. Upon their return to Earth, the barrels were rehydrated and the animals returned to their original state. Until now, it was thought that only a few types of bacteria and unicellular algae could survive these rays. Social life is not conducive to rotifers. By pure chance, though, we came across a real colony of rotifers of the Phyllodynae family. The group consists of about 30 individuals, some of which are sleeping, while others have their rotifer apparatus in action. Some Floscularia live in shells which these tiny animals construct for themselves, using small particles of detritus which they attract by moving their cilia, and which we see here swirling around this worker while he busies himself with construction. This is the empty shell of a Floscularia. The shells are of hexagonal shape, the same as those built by honeybees, wasps, and bumblebees. This, for example, is a perfect construction which I came across, luckily uninhabited. It was built by a common paper wasp inside the dome of my celestron telescope. This spectacular image is not of a peacock's tail, but that of an annelid, a marine giant, all of 3 millimeters long. Let's watch the misadventures of a ciliate which measures 40,000ths of a millimeter, and which has been harpooned by a diatom. The movement of the cilia are gradually becoming weaker and weaker, and in the meantime another diatom passes by, brushing close to the ciliate. A few seconds later, we can watch the rapid disintegration of this tiny animal, as shown in this sequence of real-time images. This is not the result of any physical laceration, but probably the effect of poison, unknown to us, which has been inserted into the victim by the diatom, and which has triggered rapid organic disintegration. This rotifer measures less than a millimeter and belongs to the Colotheca genus. It has a fan made of long brushes which it uses to bring food to its mouth and then to swallow. The banqueter's stomach is now full, and so it brings up part of the food that it has just eaten. Now let's watch the misadventures of one of its newborn offspring, a misadventure which luckily has a happy ending. Next to its mother are 5 embryos. One of them hatches and immediately sets off. It already has a set of brushes, which it has quickly started to use in order to feed. Meanwhile, the mother's own brushy fan has opened up threateningly. And the tiny newborn animal finds itself tangled up in its mother's brushes, which gradually draw it near to her open mouth, ready to devour it. But luckily the danger passes, and the little creature manages in the end to get away, albeit with some difficulty. What we see here is what we might call a multiple birth. 2 embryos are embedded in a sac, measuring about 1/10 of a millimeter. Why do we say that they are twins? The two embryos do not communicate with each other, since they are separated by an amniotic membrane. Moreover, within the sphere, the movements inside the two compartments are quite independent. Here is a ciliate which measures a few hundredths of a millimeter. Inside the cell, which is splitting, its DNA is being duplicated. It will then have two genetically identical cells. Multiplication of unicellular creatures is therefore a process of clonation, whereby each individual, during multiplication, always keeps its own genetic features. In this way, though, in the passage from one generation to another, there is no genetic variation among specimens from the same species, variations which would be necessary for natural selection and survival of the species. One way of obtaining genetic variability is through the exchange of genetic material among individuals of the same species, belonging to different families. Upon separation, neither of these two creatures will be genetically the same as before, inasmuch as they have reciprocally exchanged part of their DNA. Amoeba adopt a different approach to multiplication, simple but quick. In one part of its body, the amoeba shrinks, thereby yielding two different parts, which grow further and further apart until they split completely, as we can see in this sequence of non-real-time images. Each new amoeba also contains a new nucleus with DNA which is formed before the start of the separation process. The time required for division is less than a minute. Inside the corpses, the scavengers not only feed, but also multiply, as we can see here in the remains of this tardigrade, with pictures being taken at hourly intervals. As we shall see shortly, the birth of microscopic nematodes has a surprise in store for us. This is what the eggs that have been deposited by female nematodes look like. A few hours after the eggs have been deposited, we can see the embryos moving around, thus enabling us to see the shape that these creatures will have when they are born. One of these long annelids is carrying in its abdomen an egg, which will soon be deposited. Annelids deposit their eggs in the environment. They are always round, although they appear different. I have never witnessed their deposition, neither have I ever witnessed the development of the embryos inside the eggs, even when I kept up my observation for several days. Rotifer eggs are of various shapes, according to the family they belong to. This is a rotifer embryo inside a sac containing an amniotic-like liquid. Rotifers are nearly always born by parthenogenesis. They are all females, and only very rarely can we find male rotifers. This is a female brachionus, with two embryos that the mother is keeping close to her, while a third one is still in her abdomen. Let's watch the birth of a brachionus. The newborn creature measures around a few tenths of a millimeter. It leaves the membrane that was wrapped around it and sets off, ridding itself bit by bit of the liquid which had covered it. Here, a newborn specimen from the Colotheca family seems to hesitate before setting off into an unknown environment. This brachionus has an embryo attached to it, and another one ready to be deposited. After a short time, the first embryo hatches. It shakes itself repeatedly as it moves away, and quickly gets its cilia and mastax moving. This is the exuvia of a tardigrade, in which we can see the shells of two hatched eggs, certainly belonging to the tardigrade, as is clear from the following observations. Here we can see two more motionless embryos in the exuvia of another tardigrade. 2 days later, we can see two empty shells in the exuvia, while two newborn creatures have been found a few millimeters away, here seen under varying magnifications. One of the newborn has gone up to the exuvia which it has emerged from. Let's leave these creatures in the moss where we found them, so as to let them live their brief lives in a bit of peace and quiet. This is the embryo of an annelid. Frequent internal movement tells us that it is about to be born. Let's watch the birth. The newborn has black eyes set narrowly apart and measures a few tenths of a millimeter. These annelid embryos have been deposited in a group, and we are not able to say whether it was by one or more mothers. The births have begun a few hours after we discover them. Some of the newborn have not yet moved from their sacs, while others are already moving around. The birth of annelids holds a surprise in store for us. Some annelids have an unusual way of reproducing. One of the terminal parts of the animals breaks away bit by bit. The breakaway piece grows and is soon able to produce other individuals in the same way. This unusual type of reproduction is called binary fission. In those places where separation took place, we can see some peculiar incisures. In annelids which measure more than a millimeter, there is more than one incisure. The fission process will take place at the point where we find the incisures. This is a fragment which separated a couple of days ago. Here are two fragments of different ages. Here is a fragment which already shows sign of imminent fission. We should bear in mind that fragments are not merely simple segments with reproductive functions, as in the case of the tenure worm's proglottids, but complete animals with distinct brain and tail extremities, parts which were not present in the fission sites. We must suppose that in these places there must have been multipotent stem cells. This also explains the not inconsiderable length of time needed for the fission process. Stem cells have been used in nature for hundreds of millions of years, before they were discovered by man and utilized for therapeutic purposes. It is worth noting that annelids also produce eggs which are deposited externally, as in the case of this annelid which has laid an egg alongside. As we have already said, the class of microscopic nematodes, which are separate sex animals, holds a surprise in store for us. In addition to being born from eggs laid externally, embryos can also hatch inside the mother, where they live inside her, feeding off her tissues until they have devoured her. In this tiny nematode we can see 2 embryos which are barely moving. Let's watch the birth of one of these nematodes which, like others born earlier, will not emerge from the mother but will remain inside her. After 24 hours, it has quite clearly grown. The tissues of the mother are a reliable and abundant source of food for him. Sometimes he will push out as far as his mother's mouth until it looks like he will come out, but then he retracts. Here the mother is almost devoid of all her tissue, which has all gone to feed her offspring. Inside her body there is only an embryo, which failed to hatch. A week later, this is all that is left of this generous mother. Searching in this 8mm sea, we came across her son. As the ancient Roman philosopher Lucretius said, in order to have life, there must be death. Thus, naught of what so seems perishes utterly, since nature ever upbuilds one thing from another, suffering naught to come to birth, but through some other's death. Unicellular creatures are unstable and not very secure, and thus their chances of survival are very low, a fact which is compensated by their extremely prolific nature. If a single bacterium did not encounter enemies, it would produce another billion bacteria in one day. In a million years, the siliceous rocks consisting of an infinite number of microscopical diatoms have come together to produce sedimentary diatomaceous earth. The number of ciliates is also incalculable. The same goes for the many extremely voracious microscopic larvae, as we can see here, where they attack rotifers and their embryos. The tiny nematode is also highly prolific. This green algae is 1/10 of a millimeter long. It reproduces by means of cellular division, as we have already seen. When a number of multiplications take place in a single unicellular algae, we are dealing with a particularly rapid form of cellular reproduction, known as sporogony or multiple multiplication, erroneously called multiple division. It is the same system used by the malaria plasmodium when it multiplies inside the red blood cells of the victims of the Anopheles mosquito. Let's watch the real-time sequencing of the final moments of multiple multiplication. This strategy is not only adopted by algae, but also by some ciliates, as in the case we see here, and in the one that we will see next. This large cell, around which an inquisitive ciliate is circling, is ready to multiply. It will be multiple multiplication, as we can tell from the constant internal movement of cellular formations. Shortly afterwards, the cellular membrane breaks and a number of ciliates emerge. They soon begin to start their own rapid multiplication. They belong to the same species as the specimen which we earlier saw circling around the cell. The remains of the original cell are pounced upon by the ever-present bacteria. A hermaphrodite is an individual which possesses both male and female reproductive organs. What we see here is a half-millimeter long hermaphrodite from the marine Turbellaria family. The male reproductive organ is sticking out from the center of the creature when the female genitals is the branching terminal structure. Before the state of separate sexes was reached, hermaphroditism was the reproductive mode of many living species, and one which we can still come across in many microscopic creatures. And it is not a coincidence that in males of the human species we can find traces of Mullerian ducts, female structures, whilst in the female we find remains of Wolff's ducts, which are male features. This is testimony of our own distant and very primitive transitional phase before the sexes were separated. A cyclops hermaphrodite. The male organ is retracted. At one end we can see the branching female genital structure. In another hermaphrodite, the copulatory organ has been retracted, and we can observe peculiar movements. This hermaphrodite is busying itself spreading thousands of unfertilized eggs in the environment, grouped together in flakes. There now follows an examination of the deposited eggs. In these magnified images we can see numerous deposited eggs. Now the time has come for the hermaphrodite to fertilize the eggs, and this task takes place in a number of different stages. It then also fertilizes the flakes of eggs that have just emerged. The eggs are fertilized by an incalculable number of mobile spermatozoa. From the many fertilized cells, only a few newborn will survive. Individual chances of survival are extremely low, but just enough. This busy little animal has made its contribution to the preservation of the species to which it belongs. It has diligently carried out the task assigned to it by nature, and for which it was born. Let's wish it good luck for its rather short future life. Rotifers are born by parthenogenesis, and so most of them are female. By chance, we came across a female from the Calidine family, joined together with a small male that she is carrying with her. This is quite rare. This male is only destined to live a few more hours, that is to say, the time it takes to fertilize the female and then die. It doesn't even have a digestive tract. The meeting has been useful, however, because the fertilized eggs, containing the couple's chromosomes, will give rise to descendants which will have the genetic features of both parents. Biological evolution reached the state of the separation of the sexes after several 100 million years. In the animal kingdom, including insects, and in many species of plants, this is now the most widespread method of reproduction, inasmuch as it has proved to be the most advantageous for the variability of genetic features. Human cultural evolution has to some degree modified and even overtaken its biological counterpart. It is absolutely necessary for the deeper, authentic meaning of the latter to be rediscovered, and this message is not only aimed